Hi, welcome to NeuroScoops, where I take big concepts in neuroscience and scoop them into meaningful, understandable concepts. This process is known as chunking and is highly effective in learning and memory. Today, for NeuroScoops, I am talking about how brain cells communicate to each other through a thing called an action potential. Let's get into it. To start, we will be looking at the internal charge of a neuron. Through a bit of electrophysiology, we know the resting potential of a typical neuron is sitting at about negative 65 millivolts. Let's zoom in to an area called a node of Ranvier. This is on the axon, the part of the neuron that can send a signal to another neuron in between myelination, those blue pieces running down the axon. This is where we will be looking at for the rest of the video. Remember, the yellow part of the diagram is negatively charged and has more potassium ions. The pink part of the diagram has more sodium ions. The way we find the charge of neurons will be for another video, so stay tuned. Now that we are fully zoomed in to where we will be focusing, let's meet some of the key players, the voltage-gated ion channels. Up first, we have a voltage-gated potassium channel. This ion channel will have a conformational change when the charge on the inside of the neuron changes to a more positive charge. This ion channel is highly specific to just letting potassium ions out of the cell. There are two phases of this ion channel, either closed or open. Now let's look at the voltage-gated sodium channel. As you can see, there are three phases this ion channel can be in. First, let's look at the components. On the bottom of the voltage-gated sodium channel, we have an inactivation gate. This is a little protein that is on a ball and chain that will clog the ion channel pore to prevent the channel from letting in any more ions into the cell. Then we have an activation gate. This will open to let ions into the channel, but if the inactivation gate is in the way, ions will not be allowed to pass, thus creating three phases of the voltage-gated sodium channel. Now let's walk through the phases. First, we have deactivated. This is when the activation gate is closed and the inactivation gate is out of the way. This results in no sodium being able to enter into the neuron. Next, we have activated. This is when the activation gate is open and the inactivation gate is out of the way. This results in sodium being able to go from a high concentration outside of the cell to a low concentration in the cell. Lastly, we have the inactivated phase. This is where the activation gate is still open, but the inactivation gate is blocking the entry of sodium. This results in no sodium being able to enter into the neuron. Now let's move on to the stages of an action potential. We will start at the resting potential. This is when the cell is not signaling to other cells. Again, for the typical neuron, it will average at the charge of negative 65 millivolts. Once we get a signal that is strong enough to meet the threshold, we move into depolarization. This is when the cell is getting more positive. Once the cell reaches a certain charge, it's called an overshoot, and the cell will begin to repolarize or get more negative again. After repolarization, the neuron will undershoot and become hyperpolarized. This means it goes more negative than the resting potential. After a while, homeostasis is reached and the cell will enter into the resting potential again. Remember those voltage-gated ion channels we talked about for this? Let's see what their role is in the stages of the action potential. Starting with the resting potential, the membrane potential is around negative 65 millivolts. The voltage-gated potassium channels are closed and the voltage-gated sodium channels are deactivated. Then a stimulus comes down the axon that reaches the threshold. The membrane potential is becoming more positive, the voltage-gated potassium channels are closed, and the voltage-gated sodium channels are now activated. After the neuron has reached the overshoot phase, it will transition into repolarizing. This results in the membrane potential becoming more negative. The voltage-gated potassium channels are now open, and the voltage-gated sodium channels are inactivated. Next, the neuron reaches the undershoot phase and has become more negative than the resting potential. This results in the voltage-gated potassium channels to remain open and the voltage-gated sodium channels to remain inactivated. Eventually, the cell will restore homeostasis and will return to resting potential. The membrane potential is now around negative 65 millivolts. The voltage-gated potassium channels are closed. The voltage-gated sodium channels are now deactivated. Now let's put all the pieces together and see what is going on in the neuron. Starting back at resting potential, we can see more sodium ions on the outside and more potassium ions on the inside. The membrane potential is around negative 65 millivolts. The voltage-gated potassium channels are closed and the voltage-gated sodium channels are deactivated. Next, a stimulus comes down the axon. The membrane potential reaches the threshold. We are now entering into the depolarizing phase. The voltage-gated potassium channels are closed. The voltage-gated sodium channels are now active. This results in sodium ions to go from a high concentration to a low concentration, which means sodium ions, a positively charged ion, rushes into the cell. This causes the membrane potential to become more positive. After this, we reach an overshoot phase. This is when the action potential will transition into repolarizing. This happens because the voltage-gated potassium channels are now open, which means potassium ions will go from a high concentration to a low concentration, resulting in potassium ions to leave the cell. The voltage-gated sodium channels are now inactivated, which results in sodium ions not being able to enter into the cell. This results in the membrane potential becoming more negative. Then we enter into the undershoot phase where the cell hyperpolarizes. At this phase, the voltage-gated potassium channels are still open and the potassium is leaving the cell. The voltage-gated sodium channels are still inactivated, which means the sodium cannot enter into the cell. This results in the membrane potential becoming even more negative. Lastly, the voltage-gated potassium channels will close. The voltage-gated sodium channels are inactivated. The resting potential will be restored through other cellular mechanisms. Wow, that's a lot of moving parts, and to think our brains do this in a matter of milliseconds. If you found this video interesting, stay tuned for my next video where I start to talk about how neurons that fire together wire together. Also, if you want to explore the action potential, click the link below. I used this when I was in undergrad to grasp the key concepts of an action potential on a deeper level. That's all for now. Thanks for joining me on this week's episode of NeuroScoops. Make sure to like and subscribe to NeuroScoops and let me know what neuroscience topic I should cover in my next episode in the comments. Until next time, stay safe and be smart.